A very good morning to our precious guest, Dr. Kalpana Patel, our head of department, Dr. Chintan Tak, sir, all faculty members, and my dear students. It's my great pleasure to introduce my guru, Dr. Kalpana Patel, as a speaker of a webinar on an overview of ICH guidelines. Dr. Kalpana Patel is a vice principal, professor, and head of the Pharmaceutical Quality Assurance Department at Anand Pharmacy College, Anand. She has completed her B.Pharm and M.Pharm from MS University and PhD from Sardar Patel University. She is serving the education field for last 24 years. She has published 5 books and more than 70 research papers in various national and international journals. Ma'am has filed 3 patents related to herbal and other novel formulations. Ma'am has guided more than 72 PG students and 1 PhD student. Ma'am has delivered session on training in the area of analytical QBD and design of experiment in certification courses and theme of quality assurance and pharmaceutical analysis. She has served in various positions of honorary secretary, president, and vice president of the Indian Pharmaceutical Association. Dr. Subhas University welcomes you, ma'am. Today, ma'am will deliver a speech on ICH guidelines. Over to you, ma'am.
Thank you so much, Kathy. Am I audible? Am I audible, Kathy? Kathy, am I audible? Hello? Kathy, can you hear me? Hello? Kathy, am I audible?
Just a second.
Okay. Kathy, am I audible? Okay, okay, okay.
Hello.
Hello? Hello? Am I audible, Kathy? Hello?
Hello.
તમારો અવાજ જે
Hello,
આવે છે ને તમારો આ તમે સ્પીકરનું કાઢ
Kathy, am I audible?
Yes, ma'am. Ma'am, just a
Okay.
second. Yes, ma'am.
Okay, fine, fine. Okay.
હવે શું કરવાનું? Hello.
Hello,
ચાલો તો હતું.
hello.
Hello. Hello. થાય છે?
Hello,
કેમ આવું?
hello. Am I
અહીંયામાં
audible?
જે હતું ને
Hello, hello, Kathy.
just a second. Speaker. Yes, ma'am. You can start now. Yes, ma'am, you can start now.
Hello. Yes, am I audible, Kathy? Kathy, can you hear me?
Yes, ma'am, you can start now.
Hello. Hello, Kathy, you can hear me? Hello, Kathy.
Am I audible?
Kathy, can you hear me? હું અહીંયાથી બોલું છું. હા, એટલે સંભળાય છે ને
Yes,
તમને?
ma'am.
ઓકે, ઓકે, સારું, ચલો. I hope I'm audible now, Kathy. So
Yes.
first of all, good morning, all of you. And I would like to thank Professor Khyati and head of the department from School of Pharmacy, Dr. Subhash University for, hello, hello, hello. So I would like to thank, first of all, good morning to all of you. And I would like to thank Dr. Khyati, Professor and Head of the Department and School of Pharmacy for giving me this opportunity to share the topic of ICH guidelines. So once again, thanking Dr. Subhash University for providing me this platform for sharing the views. So today's topic is an overview of ICH guidelines. So first of all, let me share my screen. So I hope the screen is visible. So today we are going to have a presentation on an overview of ICH guidelines. So these are the contents of the presentation. That is, we will be including an overview on ICH, which includes introduction, how ICH was evolved, about the history of ICH, the organization structure, how the guidelines are developed and implemented, and what are the different guidelines that are available under different topics, and what are the ICH future goal. So as we all are aware, first of all, let us start with the ICH introduction. ICH is International Council on Harmonization, right? I hope you all know the full form of ICH, International Council for Harmonization. So ICH, particularly International Council for Harmonization, is of technical requirements for the pharmaceuticals for human use, and ICH particularly has brought together, it is an organization who has brought together the regulators, that is regulatory agency and pharmaceutical industries. And by bringing together particularly they are specifically involved in the development of ICH guidelines. And all these ICH guidelines, they are like developed on the basis of say scientific and medical aspects of pharmaceuticals. So we can say that ICH is an agency who has brought together the pharmaceutical regulators and pharmaceutical industry for the best interest of the patients for assuring global health care. So about ICH, it was like originally founded in April 1990, and ICH has grown from a small organization to a global organization, and at present it is having more than 1000 members. So major objective of ICH, what is a major objective of ICH? Its major objective is availability of safe, effective, and quality medicines, that too particularly globally, in the best interest of the patients so as to assure the global health. So at ICH, if you see, all regulators, they are committed, and they're committed for what? So these regulators, pharmaceutical regulators and pharmaceutical industry, they have come together at ICH, and they are committed and dedicated so as to promote that there should be more and more compliance for various international regulatory practices and guidelines that they have developed in various regions of ICH and non-ICH regions also. Right. So not only that, nowadays, if you know, if you are aware, like there is so much of technological advancement, right? So with this advancement in technology, innovation is a key to success. And both the pharmaceutical regulators and pharmaceutical industry, they believe that innovation is a key to success, and this innovation can be used for giving more dedicated technology in terms of healthcare with respect to patient need. So in short, ICH, which had started like 25 years back in April 1990, right? It has grown to a global organization, which is having more than 1000 members now. So particularly in a meeting in like 2015, on 23rd October 2015, under Swiss law, ICH, it was like reformed as a non-profit international organization, which is a legal organization under Swiss law. So I think you know about brief about ICH. So first of all, we will discuss why ICH guidelines are required, right? Why there was a need of harmonization, right? And how harmonization came about in terms of unified guidelines that are available, right? And today you can see on the website, you can refer the website of ICH, that is www.ich.org, right? So most of you, many of you must have gone through the website, right? So where very less guidelines were developed before this 3 decades, that is 30 years. But now if you see, there are so many number of guidelines which have been developed and implemented and framed. Why? Because of certain issues and certain problems that were raised by pharmaceutical industry. Then discussions were carried out, and then that resulted in the formation of guidelines. So all these ICH guidelines, they are available on ICH website. So before we go through those guidelines in brief, what is the need, or when there was a need of harmonization? So we all know that before 1906, right, there was no government law, right? Even if government law was there, it was not required for approval for marketing like new formulations in the market, right? So many of the formulators, they did not consider like the stringent, or did not consider in depth about the composition of the medicines, and sometimes this dangerous ingredients and excipients were also included. So if we see in 1906, Food and Drugs Act was passed, where federal government was established as a principal regulatory agency, and particularly this act was passed for like proper labeling, which emphasized on or focused on proper labeling of special drugs, and also focused on prohibiting or avoiding what the marketing of adulterated food and drugs. But only passing of this Food and Drugs Act did not serve, right? If you see the past, if you see the history, starting from 1930s to 1970s, so many tragedies, so many disasters, so many blunders occurred. Like one of the example I have put up on the slide is 1937, right? Sulfanilamide tragedy was there, where you all know sulfanilamide is a sulfonamide drug which is used as an anti-infective, right? And this sulfanilamide, it was administered to patients in the form of elixir, right, as a liquid formulation. And the formulators were not aware about the excipient which they used, and that led to the formation of ethylene glycol. And ethylene glycol was toxic, and that led to death of so many patients who had taken sulfanilamide. So that is a, like we can say in the history or in the past, the sulfanilamide tragedy which occurred, right? That led to the evolution of Food, Drugs and Cosmetic Act, which was passed in 1938. And particularly, it emphasized on that one has to, or the pharmaceutical industry has to demonstrate to FDA or regulatory agency about the safety of the new formulation or safety of new drugs. So safety aspects need to be studied in detail. Then I think all of you know about the second incident. Also, you must have studied in pharmacology, that is thalidomide disaster, which occurred in 1957 and which continued up to 1963. Particularly, it was in Europe, right? So where so many, I hope everyone knows what is the use of thalidomide. It is used as a sedative, but then the patients were also taking for the treatment of like flu and cough, and the pregnant woman used it for treatment of motion sickness. So this thalidomide, it resulted in birth of so many uh, infants, where the babies, newborn babies, were found to be born with phocomelia. That is, the limbs were not grown. Their hands, legs, that is, the limbs were not grown. Right. So again, that thalidomide. So hence it is called as a thalidomide disaster. And majority, like many of the pregnant women, used to take it for treatment of the motion sickness, and that resulted in again death of so many babies, and birth of so many babies with very short limbs. I think approximately I can say more than 8,000 birth were there. And presently also, if you see, approximately 2,000 to 3,000 like this babies who have grown up with this short limbs. So in our history, if we see, these are all the blunders, these are all the disasters. And this thalidomide disaster, particularly, it led to the passage of Kefauver-Harris Amendment Act in 1962. And that was particularly where the industry has to demonstrate to FDA before marketing that it is really safe, it is really effective enough. Right. Similarly, there are so many incidents, but I will be discussing only these two incidents, that is sulfanilamide tragedy and this thalidomide tragedy, which occurred in late 1950s. Similarly, there were so many trials also that were reported in like 1960s, 1970s. That is Tuskegee syphilis study was there, which led to the passage of the National Research Act, where a boundary was established between clinical research and clinical trial. So all these things or all these tragedies, disasters which happened previously, that led the pharmaceutical industry then at the same time, right? They were going global. Global meaning they wanted to market their new dosage forms, their formulations into international market, right? But what problem did they face? So pharmaceutical industries at the same time, they face the problem that the technical requirements for different countries were different, right? So as the technical requirements for different formulation for marketing, that is for import and export, differed from country to country, right? Although each one had to assure, the pharmaceutical industry had to assure that quality, safety, and efficacy has to be established, but technical requirements were different, and that made the process very tedious. And the pharmaceutical industry, if they wanted to market their product, say in one country, in another country, in the third country, then that cost what they had to again do retesting, again they had to submit as per their requirements, as per their specific country requirements, and they had to do various test procedures so that they could market products internationally, right? So doing all these things was like very time consuming, very tedious, and that resulted in what? Unnecessary expense. And that also led to what? Delay in introducing what? The new drugs in international market. So then, due to all this overrising cost of the healthcare and increased cost of like research and development, and delay in the availability of the formulations or new formulations in the market, right? Patient had to suffer. So then it was thought that there is a need of harmonization. Harmonization means making uniform, right? That some uniformity should be there in marketing, right? So whenever you want to market your formulation or put your formulation in international market, you have to abide the rules and regulations of that particular country. So then it was thought about by international regulators, right? That is International Regulatory Agency and international pharmaceutical industries, that harmonization should be there, that a unified process should be there, a unified guideline should be there for each and specific activity, each and every activity, so that led to what? The birth of ICH. And ICH, as we all know, it is International Council for Harmonization. So International Council for Harmonization. Harmonization means uniform, right? That is, it is same in all countries. So initially, three countries had come together. That is the Pharmaceutical Regulatory Agency and pharmaceutical industry from these three regions, that is three countries, that is EU, Europe, USA, and Japan. They formed ICH, which was called previously as International Conference on Harmonization. Then in 2015, it was renamed as International Council for Harmonization. So particularly International Council for Harmonization, it is nothing but defining technical requirements for registering the application, for marketing what? New formulation, new pharmaceutical products for human use, right? So that the data will be acceptable in any of the country, right? So ICH was incepted in April 1990 at a meeting in Brussels. And particularly the main pioneer for initiating ICH or development, major development or of ICH was Europe, right? That is European Commission, that is the EU, that is the regulatory agency of Europe. So in 1980s, they had new venture towards developing what? A single market for pharmaceuticals in the whole Europe, including say Belgium, Germany, right? And they thought about that as it was a success in Europe, they could like have harmonization globally. Globally means internationally, worldwide, right? So in 1989, at the conference of WHO held by various drug regulatory authorities in Paris, the word harmonization came into existence, and then regulatory authorities from Europe, they met the Pharmaceutical Manufacturer Association, that is IFPMA, International Federation of Pharmaceutical Manufacturers and Association, and they thought in the meeting that they should have a joint venture, joint regulatory industry initiative, so that international harmonization, global harmonization could be possible. And in that meeting, ICH was conceived as an idea. The birth of ICH took place in April 1990, where it was in Brussels, where the meeting was held in Europe, right? And that was hosted by EFPIE. So initially, representatives of regulatory agency and industry association of Europe, Japan, and America met, right? And they had formed the, we can say, the committee, right? That is the meeting. At least it should be, then they had defined that the meeting should be at least twice a year, and the place of meeting could be rotating, right, within these three regions. So steering committee was formed with meeting at least twice a year, and since then, since 1990 till date, means till today, the meetings are being convened. So as you can see, this is the mission of ICH, right? It is to achieve greater harmonization worldwide so that safe, effective, and high quality medicines will be developed, will be registered and maintained, right, with highest standards, so that they will be available in the benefit of the patients, assuring global health care. So to achieve this ICH mission, so many objectives of ICH are there. To name a few, like greater harmonization should be there in the interpretation and application of these technical guidelines and requirements which are developed for pharmaceutical product registration and maintenance, right? Then a forum should be developed for discussion on scientific issues between pharmaceutical industry and regulatory authorities, right? Particularly ICH objective is to protect the public health in the interest of the patients. Next is like ICH could develop a particular like CTD, common technical document, that is M4, and eCTD it is M8, right? And they had thought of like that was developed as a result of the mutual acceptance of research and development data, which could be accepted by each and every country as a whole. Then they had also thought for the next objective, including the therapeutic advances like gene therapy and all, and the development of new technologies. Then they used to facilitate, the next mission is to like objective is to facilitate the adoption of new or improved technical research and development approaches, right? Not only that, adequate measures were taken for implementation and integration of this guidelines and standards which are developed by giving training, by appropriate distribution, by appropriate dissemination, by appropriate communication to pharmaceutical regulators and to pharmaceutical industry. And not only that, MedDRA was also formed, that is Medical Dictionary for Regulatory Activities Terminology, right, which is a standardized dictionary, right, which is available for reference overall worldwide, right, so that any information could be shared related to the like regulatory products. So in short, ICH mission is to achieve greater harmonization globally so that safe, effective, and high quality medicines would be developed and would be available in the market for usage. So major purpose of ICH is promotion of public health, and how That is by reduction of unnecessary animal testing without compromising safety and effectiveness, to prevent unnecessary duplication of clinical trials. Say, for example, if you are performing clinical trials for marketing in India, then those same clinical trials would also serve for marketing that same formulation in another country, right? That is Brazil or any other country. Similarly, the purposes for development and manufacturing of new formulations, new medicines, registration and supervision of new medicines, and not only that, post marketing surveillance and pharmacovigilance and safety reporting in terms of adverse event. So these are the major purposes, and the major purpose, it rotates around the promotion of public health, so that the process can be made more faster, right? The process could be made more faster for registering any type of new formulation in a new market easily, right? Because the data is now acceptable internationally due to the ICH. So that's about the mission and purpose of ICH. Next is about the evolution and history of ICH. So as we know, ICH was formed in 1990, and in the first decade from 1990 to 2000, right? If you see all the timelines, the first decade was nothing but development or significant progress were there in the development of the various guidelines, and guidelines on safety, quality, efficacy, and multidisciplinary topics were developed and were emphasized for implementation in ICH and non-ICH regions. Not only those things, but CTD and MedDRA was also a major focus. So in 1990, ICH was incepted. In 1994, ICH adopted the first like MedDRA version 1, right? And where various international terminologies were included. Then first time in 1996 GCP. I hope you all are aware about the guidelines. Good practices should be there in pharmaceutical industry for assuring quality, safety, and efficacy of medicine. So three main guidelines are very important: GMP, GLP, and GCP. GMP is good manufacturing practice, GCP is good clinical practice, and GLP is good laboratory practice. So in 1996, first version of ICH E6 guideline, that is good clinical practice guideline, was launched, right? And this guideline described how to conduct clinical trials, what are the responsibilities of investigators, monitors, sponsors. I hope you all know what is the meaning of clinical trial. Clinical trial means the trial which is performed, or the experiment which is performed on humans, right? So that was the major development in 1996. Then the major development in 1999 was a formation of Global Cooperation Group, and in 2000, in late 1999, MedDRA was made available. So continuing the timeline in the next decade, in 2000, this was a major breakthrough where CTD ICH M4 was launched, and CTD is called as a common technical document, which is a single format which can be used by any country. And CTD is nothing but, it is called as a dossier. Common technical document is a dossier, and that dossier, the pharmaceutical industry in this, that particular pharmaceutical industry wants to market the formulation, has to prepare the dossier in form of a common, well structured format, that is common technical document, and that application has to be submitted to regulatory authority. But this CTD, when it was launched in 2000, it was as paper submission. But then again, it was consuming time, it was more tedious. The review by various pharmaceutical regulators was taking much time, and the process became tedious. The approval process for that particular new formulation was also not faster, was not rapid. So then in 2003, ECTD, ECTD was framed, ECTD was launched, that is version 3. So CTD in 2000, which was launched, it was as a paper submission, right, where dossier has to be submitted by pharmaceutical industry to any regulatory agency for marketing new formulation, putting the new formulation into the market. Like for example, India. So if any pharmaceutical industry wants to market a new formulation in India, then they have to submit application, which is called as CTD application or dossier, to CDSCO. CDSCO, which is a regulatory agency of India, right? So CDSCO, it is Central Drug Standard Control Organization of India. So similarly, each and every country, right, they have their own FDA, that is Food and Drug Administration. They have their own regulatory agency who will review this CTD, who will review this dossier which is submitted by the pharmaceutical industry, whether the data which is put up is okay or not, whether it is relevant or not with respect to quality, safety, and efficacy, and then they will provide their decision. So I hope it is clear that in 2000, CTD paper submissions were there, it emerged. In 2003, ICH initiated eCTD so that the process became more faster. eCTD means in form of electronic form, electronic submission, right? So electronic submissions were enabled to regulatory agency by pharmaceutical industry. So the process of reviewing by regulatory agency became more faster. So that was a major advancement, major important, significant achievement of ICH, that is the development of CTD and eCTD. Followed by that in 2088, as you can see, there are so many milestones which were achieved, right? But I'm not discussing each and every one. In 2009, the major was the ICH Q trio, that is Q8, Q9, and Q10 guidelines were launched. Q8 is particularly pharmaceutical development, Q9 is quality risk management, and Q10 is pharmaceutical quality system. So major focus was on emphasizing quality, right? So QBT approach was then transformed into QBD. Quality by testing was converted into quality by design approach. And this quality by design approach, not only that, but risk based approach. For that, Q9 guideline came up, and Q10, it is particularly for ensuring or assuring pharmaceutical quality systems are prevailing in pharmaceutical industry. So ICH Q7, that is GMP guideline, I hope everyone knows, for the manufacturing of API and formulations, right? And in 2009, ICH Q8, Q9, and Q10 came up. So second decade was simply development of new guidelines and facilitating the implementation of this ICH guidelines in not only ICH own region, but non-ICH region also. Similarly, various timelines continued with like 2011, you can see in 2012, new principles of governance were there, where the role of regulators was more defined. In 2014, Health Canada, that is the regulatory agency of Canada, and Swissmedic, the regulatory agency of Switzerland, they became the steering committee members. So since 1999, only 3 countries were there. That is 3 regions were involved: Europe, USA, and Japan. And then in 2014, 2 more were added, that is Canada and Switzerland. And in 2014, MedDRA new website was also launched. Not only that, the timeline still continued where new regulatory and industry members joined, so that ICH from a small a regional organization was converted to a global organization with more number of members in the ICH organization. ICH training started since 2017, training to various pharmaceutical regulators, that is regulatory agency, and training to pharmaceutical industry people also, that how they could adopt this ICH guidelines which came up in different sections. Particularly then in 2020, if you can see, right, the ICH YouTube channel also opened up. So particularly after, that is this last decade, since 2015, in 2015 particularly, ICH was like declared as a legal entity, as an international entity or organization, right, under Swiss law. And the major objective was to increase the international outreach, means more and more countries should join ICH, and should more and more pharmaceutical industry and should implement ICH guidelines as a whole. So these are various timelines, or how, what about the different advancements or developments in ICH since 1999 till today. Now looking towards ICH organization structure, ICH, the structure of ICH is ICH assembly, and under ICH assembly it is called as a decision making body which is assisted by two committees, ICH management committee and MedDRA management committee. And under ICH management committee, working groups are there which are called as ICH working groups for different sections, that is quality, safety, efficacy and multidisciplinary topic. So committees, and these two committees and the ICH assembly, they are particularly supported by ICH secretariat and ICH coordinator. So the meeting of this ICH assembly, right? So steering committee should be on a biannual basis, that we already mentioned, right? Along with the meetings of the management committee of ICH and Med DRA. So assembly, ICH assembly particularly, it contains all, or it is composed of all members, and particularly they take decision regarding the framing of guidelines, admitting new members in ICH, then defining the procedures, defining rules, right? After framing ICH guidelines, this ICH guideline should be adopted by pharmaceutical industry. So these are all the roles and responsibility of ICH assembly. And this ICH assembly, it particularly takes decision by consensus. What do you mean by consensus? By agreement of all regulatory members. And if there is disagreement, then voting is done, where only regulatory members have the right to vote. Right. So that is about the ICH assembly. Under ICH assembly, we already saw that management committee is there. And this management committee, it is responsible for monitoring the operational aspects of the association or organization, including both administrative and financial matters. And not only that, management committee also provides recommendation that which new topic should be selected for harmonization or for preparation of new guideline, or which guideline requires amendments or changes, or which guideline should be withdrawn, right? So all these things are discussed by management committee and they make recommendations to ICH assembly. So particularly ICH has two types of people, that is members and observers. 20 members are there and 35 observers. 20 members include, as you can see on the slide, founding regulatory members, founding industry members, standing regulatory members and industry members. The founding regulatory members are Europe, USA, and Japan, who had initiated ICH in 1990. And the founder industry members are EFPIA, that is the association, European Federation, Pharmaceutical Industries and Association of Europe. Similarly, for Japan, it is Japanese Pharmaceutical Manufacturer Association, and for USA, it is PHRMA, that is Pharmaceutical Research and Manufacturers of America. So founders means who had started ICH, right? Who were involved in the starting of ICH. So 3 countries, Europe, USA, and Japan, their regulatory agency and industries, where industry and associations were involved. Then as the time passed, the total number of members increased from 3 to 20 at present. So standing regulatory members are Canada and Switzerland. Health Canada is the regulatory agency of Canada, and Swiss Medic is the regulatory agency of Switzerland. And there are, you can see the regulatory members also, that is Brazil, Mexico, Singapore, Korea, UK, China, Saudi Arabia, TEPI, Turkey, and 3 industry members, that is Bio, Global Self-Care Federation, and IGBA, right? So these all are 20 official members. And the second type of people who are involved are observers, that is 2 standing observers and 33 observers are there. So 2 standing observers are WHO and IFPMA at international level, and 33 observers include the various regulatory agencies of 20 countries, which you can see on the uh, slide, that is Azerbaijan, Argentina, Algeria, India, Cuba, right? Then 6 harmonization initiatives of the particular region, like Asian region, EAC, that is East African Community, similarly GHC, PANDRH, SADC, APEC. So these are all 6 regional harmonization initiatives who have joined as observers. Then EPIC, that is the International Pharmaceutical Industry Organization, and 6 organizations who are implementing and regulating this ICH guidelines, which includes the Bill and Melinda Gates Foundation, CIOMS, IDQM, IPEC, right? And the particularly PICS, that is Pharmaceutical International Convention, and USP. So 35 observers and 20 members, right? They make up the ICH organization. Members are called as the regulatory members, who are involved in development of the guidelines and can take decision, while observers can attend ICH assembly meetings, but they don't have any right to vote. And there is limited eligibility criteria for new observers, and particularly no duty it is like given to any observer, but any observer, that is to WHO and IFPMA, they have the right to appoint the experts in working group. Right, so there are 2 types of working group in ICH, that is EWG and IWG, that is implementation group and external. So as you can see, this is just for showing you the facts and figures that ICH members and observers since 1990 to 2021, they have increased, right? And right now more than 1,000 individuals are involved in ICH work, right? Particularly 708 experts in different 34 working groups are available as of May 2022. That is a statistics which has been released by ICH. So any pharmaceutical industry or any regulatory agency, can they become member of ICH? So the eligibility criteria is that yes, they should be involved in ICH process. They should have attended uh, any attendance or any meeting in the past in ICH, or certain experts should be appointed in the working group, and ICH guidelines should be also applied in those pharmaceutical industry. Then and then they can become member in ICH assembly. And particularly the regulatory agency, they should be having the like implementation aspect of Q1, Q7, and E6, which are the major guidelines. Q1 is stability testing guideline, Q7 is GMP, and E6 is GCP. Then and then they can become member. And particularly industry point of view, the pharmaceutical industry should involve in the adoption and implementation of this ICH guidelines. Then and then they can become member in ICH assembly, right? So any industry or any regulator cannot become member in ICH assembly, but they have to fulfill the eligibility criteria. So then how the process of harmonization is there? It is particularly classified into 4 categories: formal ICH procedure, which is nothing but development of a new guideline; then Q&A procedure, that is question and answer procedure, that is creation of question and answers so as to assist implementation of the existing guidelines. So it is a sort of FAQs. Then the third category is revision procedure. Revision procedure means any new guideline is launched, then after certain period of time, certain industry people, they are facing problem or issue in adopting that guideline or in execution of that guideline, then certain revisions or modifications can be again suggested. So revision procedure is also possible, that is where revision or modification of the existing guideline can be there. And the fourth category is the maintenance procedure. And maintenance procedure, where to the existing guideline you can add some more standards or recommendations can be added. So process of harmonization, particularly, I hope you all know, it is divided into 4 categories: formal ICH procedure, which involves development of a new guideline; QA procedure; revision procedure; and maintenance procedure. So internal working group, the implementation working group, right? It is particularly involved in the second category procedure, that is question and answer procedure, while the EWG technical discussion group, it is involved in rest of the 3 procedures, that is formal ICH, development of new guideline, or any revision should be done, or some standard should be added to the existing guideline. So in that, EWG external working groups are involved. So normally for development formal ICH procedure, it is related to development of new guideline. So new topic should be first of all identified, and that should be harmonized by ICH. And for that, first of all, concept paper has to be built, business plan has to be required. So if you visit the ICH website, concept paper and business plan, they are put up, which are nothing but the objective, right? Why that particular topic requires harmonization, right? Or uniformity should be there. Okay. Similarly, for Q&A procedure, concept paper is required, and clarification it is needed for the existing ICH guideline. Similarly, concept paper it is required for revision and maintenance procedure also. And for revision, content of existing ICH guideline, say if it is no longer valid, or say it has become out of date due to certain like technical advancements or due to significant advancement in science, then revision procedure should be adopted. If there is some new information, then that should be added to the existing guideline. That is also included in revision procedure, right? And certain maintenance procedure where recommendations are there in the existing guideline, that can be done in maintenance procedure, where concept paper is required for maintenance of guideline, but no concept paper is required for recommendations. So this is how this four categories, the harmonization, it is divided into four categories, and ICH guidelines, how they are developed. So these are the steps in the process to select like new topics for harmonization, where all ICH members and observers, first they will identify that yes, for this particular topic, a guideline is required. So all ICH members and observers will submit the topic proposal for the new topic. Then this topic proposal will be put up to ICH management committee. This ICH management committee will review the new topics which are proposed by members and observers. Then recommendation will be given to ICH assembly. ICH assembly will then give a decision on topic and will establish a particular working group based upon the topic and experts related to that particular topic. Then further, concept paper and business plan will be developed by the working group. Again, that will be submitted to management committee of ICH, and that management committee will oversee or review that particular concept plan and business plan and will execute for adoption. And then the information will be passed on to ICH assembly for establishment of expert working group or implementation working group. So this is how a new guideline emerges in in ICH, right? That how the process is being followed, and the meetings are normally, as we saw, biannually. It is conducted particularly in November and June. ICH assembly meetings are there. So as you can see, these are the process, 5-step process in the development of the guideline, with the working group prepares draft of the technical document. That is the document is prepared, a technical document is prepared. Then the members of ICH assembly, then they are invited to see this technical document. They will review the technical document, and then after finding it suitable or appropriate, they will endorse it that it is proper. And then the regulatory members who are involved in the ICH assembly, they are invited to draft a guideline on the basis of this technical document. After the new guideline is prepared or drafted, in step 3, it is available to the public in the public domain, right? Where various comments are invited, and these comments are considered, each and every comment which is received, right, by the ICH regulatory members and ICH, not only ICH regulatory members, but the public also, right? Where different pharmaceutical industry and regulatory agency can provide their comments, right? These comments are considered by the working group, and finally the agreement is reached that yes, agreement is reached among whom? The working group, right? That all these comments which have been received, right, they try to solve it, they try to incorporate it in the guideline, right, in that particular new guideline, and then they finally prepare the guideline. After step 3, step 4 is there, that is adoption of final ICH harmonized guideline. So finally, that ICH harmonized guideline, it is an adopted by the regulatory members of the ICH assembly, and then step 5 is implementation by the ICH regulatory members. So this is how a new guideline is being drafted in 5 steps, starting from step 1 to step 5, starting from preparation of the technical document till implementation by ICH regulatory members, and then this guideline can be also adopted by non-ICH members. So particularly if you have visited the ICH website, there are 4 broad categories of guidelines. That is, you can see those blue letters on ICH website, that is Q, S, E and M. Q is quality, S is safety, E is efficacy, and M is multidisciplinary. So quality guidelines, they're related to chemical and pharmaceutical quality assurance for assuring quality of the formulation. So for assuring quality of the pharmaceutical formulation, you need to perform what? Stability studies. You need to assure impurities. If impurities are present, then that has to be tested whether it is present in the acceptance criteria or not. Right? Pharmaceutical quality system should be there. Right? GMP principles, that is good manufacturing practices, has to be implemented. So first section is quality, right? Second category is safety. Safety involves mainly in vitro study and in vivo preclinical study. In vivo preclinical study meaning studies which are done or experiments which are done on animals, right? Not only that, various testing including carcinogenicity, genotoxicity, that has to be performed for assuring safety of a new formulation which has to be put into the market. The third broad category includes efficacy, which relates to clinical studies in human. So clinical studies in human, where you have to adhere to good practice, which is called as good clinical practice, which is specifically for designing clinical trial, for conducting clinical trial as per ICH E6 guideline, right? And safety reporting and documentation of clinical trial should be also done as per ICH E6 GCP guideline. In efficacy, not only this pharmaceuticals which are explored for clinical studies, but it also includes various novel medicines based on biotechnological process, that is DNA based medicines, right? The use of pharmacogenetics, gene based therapies, pharmacogenomics techniques, right? Which are there, gene based therapy for targeting or targeted delivery. So efficacy of all this type of not only pharmaceuticals but advanced therapeutic should be also assured. And last category is multidisciplinary. So any topic which does not fit in Q, S, and E, right? They are included in multidisciplinary topics, including MedDRA, ESTRI, CTD, ECTD. So here we will go through the guidelines where you can see that I have put up various quality guidelines. So as you can see, it is from Q1 to Q14. Q1, it is specifically focusing on stability testing, which includes from Q1A to Q1F. So I will explain meaning of only one. That is, what do you mean by Q1AR2? So what is the significance of Q1AR2 guideline? Q means quality. 1, it indicates the word stability topic, right? 1 is stability topic. A means stability testing. It is done for what? For new drug substance and new products, right? And R means revision, and second time that revision has been done, right? So you can see that only in certain cases or in certain guidelines that bracket revision has been mentioned. So Q1B, Q1C, Q1D, all those are the original guidelines which have not been revised. So if the guideline is revised one time, then you have to denote it by symbol R1. If it is revised second time after some years, then you have to denote it by symbol R2. So that is the significance of writing guideline, right? That is Q1AR2 means stability testing of new drug substance and product which was launched originally has been revised twice over the period of years, right? Similarly, you can see for Q1B, C, D, E, F, photo stability testing guideline is there, right? New dosage form guideline is there. Bracketing and matrixing design is available for stability testing. Then evaluation of stability data, right? Then Q2 is analytical validation. So as you all know, like for new formulation and for existing formulation, like stability programs are there, which are included in Q1 guidelines, stability testing guidelines. Similarly, for performing assay, I hope everyone must have performed assay, right? Which is available in pharmacopoeia, either IP, BP, JP, USP. So for performing assay, you require analytical methodology, and the analytical method needs to be validated. So for validation of this analytical method or validation of analytical procedure, Q2 guideline is there, which includes various validation parameters, including like accuracy, specificity, precision, linearity, range, right? LOD, that is limit of detection, limit of quantitation, sensitivity, selectivity, robustness, ruggedness. So validation of analytical method, it is very important. Why? Because that analytical method, it is used for checking the assay, or it is used for performing what the assay. So this testing methodology or analytical method which you are using in laboratory should be so much accurate that it should assure the quality of formulation. Analytical method, it is used for assay, meaning that it is assuring the quality of formulation. Similarly, Q3 guidelines are there. So right now, if you see Q2 guideline, which is available as analytical validation guideline, that is R1. Right now, it is under revision, that is Q2 R2, and a combined guideline has been launched, Q2 R2 and Q14, right? Combining analytical validation and analytical procedure development. Because previously in Q2 R1, it was including only validation, analytical procedure, that is optimization of analytical method, how you are developing that analytical method, that aspect was not included. So Q2 is analytical validation. Q3 guidelines are focusing on impurity topics, right? A, B, C, as you can see, A is for new drug substance, B is for new drug product, C is for residual solvent, D is for elemental impurity, and E is for extractable and leachable for pharmaceuticals and biologics, right? And that is under development. So I hope impurity also everyone is aware about. Impurity means the unwanted chemical which should not be present in the formulation in the API, right? And even if it is present, it should be present in a particular level, or a particular acceptance criteria should be there, which is nothing but different threshold levels are indicated in the guideline, reporting threshold, right? So there are different type of thresholds that you can go through. Then Q4 is pharmacopoeia, where pharmacopoeial harmonization is there, particularly USP, and Q4B is including various pharmacopoeial tests, right? Text, and various tests are given that how it should be performed, and various annexures are available, as you can see, which are listed on the ICH website, starting from Q4B annexure 1 to so many annexures, that is 13. Okay, and that is particularly for like extractable volume of parental preparation, how you can perform that particular test. If you wanted to perform for particulate contamination, then you can refer how you can perform microbiological examination or test for say non-sterile products, right? So all these details are available in this guideline, right? In the pharmacopoeial guideline. Q5 is particularly focusing on topic of biotechnological product and assuring its quality. So A to E guidelines, 5 total guidelines are available under biotechnological product, right? And as you all are aware, biotechnological product is derived from cell line, either from the animal or human origin. So the safety evaluation in terms of like viral or virus should be assured, right? B is the cells which are used. So what is the gene expression study, and all those things should be assured. C is particularly related to stability testing of biotechnological product, because stability program for biological or biotechnological product, it is different from pharmaceutical formulation, right? So these are all the guidelines. Q5 is focusing on biotechnological product. Q6 is including specifications, that is Q6A and Q6B. So I think you are all aware about what you mean by specification. So if you have referred IP Indian Pharmacopoeia, specification means acceptance criteria. Whenever you perform any assay, it will be written that the result should be like, for say, if you're performing assay of paracetamol, assay should be between 95 to 105 percentage, 105 percentage. So this is called as the range, and that is called as a specification or acceptance criteria. So Q6A are nothing but how you can decide this guideline. It includes what? Q6A specification guideline includes that how you can decide acceptance criteria for new drug substances and for new drug products. And Q6B specification guideline mainly focuses on the how you can fix the specification limit or acceptance criteria for biotechnological and biological products. Similarly, Q7 is GMP, that is Good Manufacturing Practice Guideline. Q8 is Pharmaceutical Development. Q9 is Risk Management. 10 is Pharmaceutical Quality System. 11 is Development and Manufacture of the Drug Substances, including both entities, that is pharmaceutical chemical entities and biotechnological and the biological entity, right? Q12 is life cycle management for the formulation. So what are the technical and regulatory requirements for the whole pharmaceutical product life cycle management? All these aspects are included in this guideline. Q13 is it came up recently only, and before few months, that is continuous manufacturing guideline, particularly for both drug substance and drug product, that how continuous manufacturing can be enabled, that has been explained. And Q14, which is still the expert working group is still working upon, right? That is analytical procedure development aspect and revision aspect, right, of analytical validation. So these are all the 14 quality topics, or you can say quality guidelines, which are available. Similarly, safety guidelines are available. So safety guidelines, that is S1 meaning carcinogenicity study, S2 is genotoxicity study, S3 A and B. A is toxicokinetic study and B is pharmacokinetic study, right? Chronic toxicity testing, that is S4, right? S5 is reproductive toxicology study. Then toxicology or preclinical safety evaluation of biotechnological product, that is S6. Then pharmacology studies, right? Safety pharmacology or safety aspects in terms of the human pharmaceutical non-clinical evaluation, right? Both these things are included in pharmacology studies, that is S7A and S7B. Similarly, S8 is there, S9, that is non-clinical evaluation for particularly anti-cancer pharmaceuticals. S10 is photo safety evaluation. S11 is non-clinical pediatric safety studies. S12 is non-clinical biodistribution consideration, right? In the biological system, which should be considered for particularly gene therapy product, right? So these are all the 12 different types of guidelines which are available. Efficacy guidelines includes even clinical safety guidelines for drugs which are used in long-term treatment. E2, 2, that is from A to F, different pharmacovigilance aspects has been included. E2 guidelines, that is efficacy guidelines which are performed on, I hope you remember what is efficacy. Efficacy means study on humans. There are different type of efficacy guidelines. E1 is clinical safety for drugs. E2 includes all pharmacovigilance from A to F. Different aspects of pharmacovigilance are included, like example, clinical safety data management or individual case safety reports, ICSR, right? Then benefit risk evaluation report has to be prepared. Similarly, E3 guideline is there, E4 dose response study is there, E5 focuses on ethnic factors, right? And how this acceptability will be there with respect to the foreign clinical data. E6, as we discussed, it is GCP guideline. E7 is clinical trials to be performed in considerations for geriatric population, right? Older age population. E8 is general consideration for clinical trials. So E6 and E8 go along. E9 is statistical principles which should be adopted for the clinical trials, and E10 is choice of control group, how you should select control group in clinical trial. E11 is particularly clinical trial which should be performed in pediatric population, that is children. E12 clinical evaluation which should be done by like various therapeutic category, like antihypertensive category or like that, right? E14 is clinical evaluation of the QT, that is evaluation of QT, QTC interval, right? Then pharmacogenetics, pharmacogenomics, that is E15, E16 is qualification of genomic biomarkers, E17 is clinical trial which should be considered with respect to more than one region, that is multi-region. E18 is genomic sampling, right? And management of how this data of genomes will be stored and used, right? E19, safety data collection related to clinical trial, and E20 is adaptive clinical trial, right? So in safety guidelines, you can see that there are 20 topics, right? And next is multidisciplinary guidelines, that is M guidelines, M1, M2, M3, M4, M5, right? MedDRA terminology, which we already discussed. M2 are electronic standards. M3 is non-clinical safety studies. The major important development was M4 and M8, that is CTD and eCTD, common harmonized format which is available, right? Then M6 is gene therapy, M7 mutagenic impurity guideline, M8 is electronic, that is eCTD which we discussed, M9 is BCS based biowaiver, M10 is bioanalytical method validation guideline, M11 is the clinical electronic structured, the harmonized or unified protocol, M12 is drug interaction studies, M13 is bioequivalence studies, M14 is how real world data can be used in pharmacoepidemiology study. M15 is how general principles are to be applied for model informed drug development. So these are the 15 subtopics, 15 topics which are included under multidisciplinary guidelines. So I hope you got an overview related to the quality, safety, efficacy, and multidisciplinary topics. Each and every guideline, it is available in detail on ICH website, which you can go through, right? So the major important thing which we discussed previously also, that is ICH standard, the common technical document, CTD, eCTD. So how it is unified, because it is a common format, you can see in the form of the triangle. So module 1, module 2, module 3, 4 and 5. These are 5 parts of the triangle. This is the module 1 is administrative information. So that differs from country to country. So say for example, India. So for India, the forms of application and everything, the fees requirement and that will be different. Say from USA, from Europe, right? So module 1 is administrative information which differs from country to country, and module 1, it is not considered as part of CTD, but the 4 modules, that is module 2, 3, 4, and 5. They are considered as part of CTD. Module 2 is particularly summary, which includes summary of the quality information, the non-clinical overview and summary of non-clinical studies, overview and summary of clinical studies. And module 3 is specific quality aspect, which includes chemistry, manufacturing, and control aspects, that is CMC information. Module 4 is non-clinical data or non-clinical study reports which are put up in the dossier. And module 5 is in detail clinical study reports which are performed as clinical trials on human. So CTD triangle just divided into, which includes the 5 modules. Module 1 is administrative, which is region specific, but module 2, 3, 4, and 5, they are considered common for all countries. So CTD became what? Since 2003, CTD became the mandatory format, right? So for preparing what? The application, right? For putting up any new formulation in the market. So quality, safety, and efficacy. Q is module 3, that signifies quality. Non-clinical study reports in module 4 specifies safety, and module 5, it focuses on efficacy. This is just to show you that ICH, how ECTD was implemented, and this shows that in majority of this, like 3 ICH regions, Europe, USA, and Japan, it is mandatory to put up the dossier or application should be submitted in ECTD format only, that is electronic version, right? These are other ICH standards, MED-DRA, which is particularly used for registration, documentation, and safety monitoring for medical products, like both before and after they are available in the market. Then various other electronic standards are also there, that is ESTRI, electronic standards for transfer of regulatory information. Then we discussed about M8, right? E2B is also important, which is nothing but the electronic transmission of ICSR, that is individual case safety reports, which are prepared in clinical trial particularly. So besides availability of the guidelines, there are also reflection paper. So reflection paper is nothing but where you identify, the members and observers identify a new topic for the preparation of a new guideline, and they put up on the paper, which is called as reflection paper. And on the basis of the reflection paper, discussion groups are formed. And this discussion groups, right, it is nothing but a technical forum, right, who invites experts from that particular area for which new guideline has to be prepared, right? And they prepare. So if you see on the website right now, all these topics are new topics, that is model informed drug development, patient focused drug development, the pharmacoepidemiological studies requirement, harmonization for standards should be, harmonization of standards should be there for particularly generic drugs which are available in the market, and GCP renovation, right? So discussion groups, they're particularly formed on the basis of the reflection paper. The next consideration documents are there. Consideration documents are different from guidelines, and these documents are prepared by discussion groups, particular discussion group. So that does not, this consideration document does not require approval from ICH, but just the required discussion in the meeting, and then ICH assembly endorses that particular consideration document. Right. About stability testing guidelines. So I hope you are aware now about what is ICH, how ICH has been formed, right? About the organization structure of ICH, how ICH guidelines have been formed and implemented. What are the different ICH guidelines which are available under quality, safety, efficacy, and multidisciplinary topics. And the heart of the most important parent guideline which is available is stability testing guidelines. So we will just hurriedly go through this stability testing guideline, because uh, stability program is necessary for if you want to put up any new formulation into a market. So unless and until you assure stability in terms of physical, chemical, microbiological, and biological testing, if all these things are like approved, right, with that formulation is physically stable, chemically stable, microbiologically stable, or biologically stable, then and then you can put up that particular formulation in the market, right? So these are the different guidelines. Stability, as we all know, is capability of any formulation or particular formulation to remain in its physical, chemical, microbiological, and therapeutic and toxicological specification in the specific container closure system. So there are three types of mainly stability studies: long term, intermediate, and accelerated. Long term means the duration is more, intermediate means it is intermediate, and accelerated means the conditions are stress driven or are having higher accelerated conditions, that is forced conditions are used. So looking towards the first guideline, we're not going in more details, but the guideline was revised 2 times, and purpose of stability testing particularly it is to assure that how the quality of the drug substance, that is API, or drug product, that is formulation, changes with the period of time, over the period of say 1 year or over the period of 2 years, right? How environmental factors are going to affect, like temperature, humidity, light, right? So this is just to show you the progressive changes in ICH guideline. 2 times revision were done, right? So in Q1AR accelerated condition, there was change, that is instead of 5 time points 0, 1, 2, 3, and 6, it was only 3 point study. Similarly, progressive changes in stability storage condition, right? That is in the second time revision when it was done, it was recommended in long term and intermediate stability storage condition, that is either the applicant can perform studies, stability studies at 25 or 30 degrees centigrade, right? And 60 or 65 percentage relative humidity, that is plus or minus 5 percentage related plus or minus 5 percentage in relative humidity, and plus or minus 2 degrees centigrade temperature, right, with respect to temperature. So there are, I think everyone knows, there are 4 global climatic zones. On that basis, stability testing program has to be designed, right? 4 zones including moderate, Mediterranean, hot and dry, hot and humid. And on that basis, the temperature and humidity has been enlisted. So in stability testing, major considerations are with respect to the selection of batch, what general information, container closure system. So all these things, the important things I have put up. That is, first important thing is selection of batches. So stability program should be performed on minimum 3 batches, 3 manufacturing batches, and that could be from the pilot scale or plant scale. And these 3 batches which are manufactured should be manufactured by same manufacturing process, by using that same equipment and by using same route. Then and then you can select those batches for performing stability program or stability testing. Similarly, container closure system, everyone knows, in which the drug product or the formulation is proposed for storage and distribution and will be available to the patient, right? So stability should be also assured in container closure system, right? Testing frequency, it is third important aspect of stability program or stability testing. And testing frequency is also again classified into long term, intermediate, and accelerated. And long term testing frequency includes, like for first year, every 3 months you have to carry out testing. Should be the frequency in the first year, then 6 months frequency, that is 2 times in the second year, and in the third year, thereafter it should be annually, third or fourth year. Intermediate includes 0, 3, 6, 9, and 12 months testing, instability testing program, and accelerated testing is 0, 3, and 6 months. So these are all the different important criteria, that is selection of batches, container closure system, the stability program should be performed, testing frequency should be defined, then storage condition. Storage condition is like very, very important, like general cases defined in the guideline for drug products, right? So long-term, intermediate, and accelerated studies. So how much should be the storage condition, that you can see in the particular guideline, long-term 25, intermediate 30, and accelerated 40, along with the humidity 25, 60, 30, 65, 40, 75. And the duration is also mentioned. So if any significant change it is occurring, what do you mean by significant change? Significant change means, say when you carry out any stability testing, say you are carrying out stability testing where the attribute is assay. Attribute means in stability testing, what you are going to check, what you are going to verify. Say if it is a tablet formulation for which you are performing stability studies, right? Then for tablet formulation, it can be assay, right? It can be dissolution study, okay? It can be disintegration study for which specific acceptance criteria or specification limit is defined. So say for example, for assay, if specification limit is defined, then in long-term study, you will carry out at 25, 60 combination respectively for temperature and humidity, right? And the long term means the duration will be 12 months, intermediate the duration will be 6 months, and accelerated the duration will be 3 months. And see if you are, I was giving you example of assay. So if you performed assay, then if you have the acceptance criteria, right? So if your result is within the acceptance criteria, then it will not be called as what? Failing, right? It means that always you write down the result as what? That it is passing the acceptance criteria, or it is passing the that particular specific specification criteria because it is in that particular limit, right? So there is no significant change in the formulation, right? There is no changes or no variability in the formulation has been found during the stability testing program, right? So your result will be the same. Okay, so if your result is same, right, over the period of say 12 months or 6 months, then you can say that there is no significant change. But say in accelerated study, if you are performing and if any significant change is found, then you should carry out additional testing in intermediate storage that should be conducted. So then it has to be extended, right, from 6 months data to 12 months data for intermediate storage condition should be considered if in accelerated stability testing significant change is observed. If it is observed, then you have to include what? Minimum of 6 months data in the initial application. The study can be extended up to 12 months, right? Because significant change was found. Similarly, for stability testing for drug substance or drug product, you can see refer the guideline Q1A, B, right, where it is recommended for storage in refrigerator, recommended for storage in freezer. So these are the conditions which are listed. This is for just showing you what you mean by significant change. So as I told you, significant change means failing to meet the specification. If the specification is not passing, if it is not within the prescribed or defined results, it is called as failing to meet specification. Okay, so that is called a significant change. And for formulation, if there is say 5% assay variation from its initial value, it will be called as significant change. If any degradation product is forming, right, which is not remaining in its acceptance criteria, that will be called a significant change, right? If certain physical attributes are changing, like color, appearance, hardness, pH of the formulation is changing, right, then that will be called as failure to meet the acceptance criteria. But sometimes when you carry out the stability testing in accelerated condition, if the formulation is suppository, right, then softening of suppository might occur. If it is a cream formulation, then melting of cream formulation may occur, right? But that is then expected at accelerated condition. So such factors should be taken into consideration while you are designing what stability testing, when you're performing stability testing, and when you're evaluating the stability testing results. This is just to give you stress testing overview. It is particularly performed on single batch of drug substance. Stress testing means you are performing study at higher temperature, higher humidity. Higher temperature means you will add on 10 degrees centigrade increment, like say 60 degrees centigrade, 70 degrees centigrade, 80 degrees centigrade. You will use what? Accelerated condition, that is forced degradation condition, right? And then you will check whether the API is stable or not, right, under this accelerated condition, whether it is undergoing hydrolysis or not, whether it is undergoing oxidation or not, right, whether it is photostable or not, right? So photostability testing, it is also integral part of stress testing, both for API and formulation. So stress testing, what is the importance of stress testing, whether it is drug substance or drug product? It helps to identify the degradation pathway, right? We can know the stability of the API, whether it is stable under acidic condition or not, alkaline condition or not, in presence of moisture, that is environmental factor, which is very important, right? How oxygen, which is present in the environment, will be having impact on the stability of API, right? What analytical procedures which will be used for stress testing. So these are all the things which should be considered during stress testing of drug substance and drug product. Then evaluation. Evaluation as a best case means the stability testing. So when can you say that stability testing has passed or stability program is a success? When no significant changes observed at accelerated condition within 6 months. Long-term data, it shows very little or no variability, or very little or no change over the period of time. If accelerated data, the results which you have accumulated in accelerated studies show little or no variability or little or no change over the period of time, right? And if statistical analysis in stability testing program is say not necessary on the data which has been gathered, right, then the proposed retest or shelf life period will be double of the period covered by the long term data. So long term data is normally, long term studies, it's normally performed for 1 year, 12 months. So in that case, retest period or shelf life will be double of period, meaning 2 years, right? So I hope everyone knows about shelf life. That is uh, the expiry date which is available on the formulation package, which you must have seen, right? So shelf life, it is nothing but the duration or the time period in which the API or the formulation remains what? In unchanged condition or within the specification. It remains within the specification. And retest period means it is a retest date, and after this date, you require the API or the formulation to be tested. So that testing is required, so that you need to assure that whether it is within the specification or not. That is the meaning of retest period and shelf life. So retest period or shelf life can also be granted on the basis of the results of stability program or stability testing by extrapolation method. But extrapolation method, when it is used, right, it should be always verified by long-term stability results data. So related to extrapolation in the Q1, that is E guideline, which is evaluation of stability data. What is the meaning of extrapolation? And that has been given in detail, right? Next is variation. Variation means when once a formulation is available in the market, it might happen that certain changes are there in the manufacturing process, or changes are there in the composition of the formula of the finished product, or changes in the packaging material, or changes in the manufacturing process of an API, or changes in the selection of the vendor who is supplying the API. So wherever there is variation, it means what? Changes. Then again you have to perform the additional stability studies. So it is not the case that only once you wanted to put the formulation into the new formulation into the market, at that time only you require stability testing or stability program. It is not that. So after it is available in the market, that is after post marketing also, right? After post marketing, that is after it is available in the market, if certain changes are being done by the pharmaceutical industry or company in the manufacturing process, or some excipient, or some manufacturing process of API or packaging material, then again stability studies has to be performed by that particular pharmaceutical industry to assure that it is not causing any variation in stability testing results, right? Then this is just to give you the glimpse on photostability testing, right? Where photostability means how the light, UV light and visible light, it is going to affect the formulation. So directly this photostability testing study, it is performed on API, that is drug substance. It is also performed on the drug product outside the packing. It is performed in the immediate packing also, and it is performed in the formulation in the marketing packing also, right? So all this type of photostability testing studies, it assures, right, or ensures that yes, the drug, it is not getting degraded in presence of light, or it is not affected by the UV and visible light. So these are the two light sources which are referenced in the guideline, which you can refer. Similarly, for drug substance, you can perform what? The post degradation and the confirmatory testing study, right? In photostability testing, particularly for evaluating the photosensitivity, either it can be performed in solid form or in liquid form also. It can be performed in the container closure system also. These are all the conditions which are listed in the guideline. This is the decision tree which is listed for, or it is shown in the guideline for photostability testing of new drug substance and products, right? Where directly the formulation is exposed to light. If the change which has occurred is acceptable, then you have to end the test. But if it is not acceptable, some like if you expose that formulation, then what will happen? If you expose a formulation and if it is not acceptable, then you need to go to the next test. So you can refer the decision tree, right? Then stability testing for new dosage form guideline is also there. For new dosage form, meaning say different administration route or specific delivery systems are available. Then bracketing and matrixing design is also there for stability testing. Bracketing and matrixing, it is particularly used for stability program when different strengths are there, different container sizes are there, right? So in the blue color, you can see an example of container size ki 1500 and 500 ml, and containers are available which are packing 50 mg, 75 mg and 100 mg. So bracketing means the extremes. Extremes means the 15 ml and 500 ml, right? For 50 mg and 100 mg has to be tested, right? Has to be tested. So stability of the extremes have to be performed. That is a designing. Similarly, for matrixing also, it is called as a designing of the stability schedule, where at different time points, all samples will not be tested, but Selected samples will be tested, say, for at the third month, and selected samples will be tested from that particular stability program which is going on at the 6 months, and then the results will be accumulated. So, for example, this is to show you how matrixing is done for two strings, that is S1, S2, right, for different batches, right? So, as you can see, for 0 and for 3 years, all the batches have been tested. But if you see for 3, 6, 9, 12, 18, 24 months, right, for batch 1, 6 and 18 months is skipped for strength 1, and for batch 1, for strength 2, 3 third month has been skipped. So that is called as the matrixing design, right? The evaluation of the stability data that we have already talked about, that is significant change. Then we have Q1E guideline also, particularly for framing or deciding the shelf life of the formulation. Shelf life means expiry date, and that depends upon what the long term and accelerated data. So significant change, if it is there or not, that you have to pose the question in accelerated condition. If it is no, then you have to follow this steps, and if it is yes, if significant changes yes, then you have to follow these steps, right? And then based upon the studies, you have to define the retest period or the expiry date, that is shelf life of the formulation after performing the whole stability studies. This is just to show you that for different formulations or for different dosage forms, which all physical parameters are evaluated, like for solution, suspension, tablet, gelatin, capsules, emulsions, creams, right? This is a guideline for climatic zone 3 and 4, where again, product shelf life, it is defined based upon the climatic condition of zone 3 and zone 4. And this type of studies, like accelerated and long-term studies, are specifically performed at this specific temperature and relative humidity for different type of formulations, that is oral dosage form, glass bottles, right, drug products. So coming to the end of the presentation, right, what are the key takeaway or keys to the ICH success? That ICH as an international agency, it has grown from a small organization to a large organization. And nowadays, if you see the ICH website, in all these topics, including Q, S, E, and M, all the guidelines which are available, they are all like science-based guideline, right? And the process is also effectively managed, right? And the regulators are very much committed and have included innovation aspect also, so that products can be developed with innovation and that can be harmonized, right? And in the better interest of the patients, for assuring global health, it can be made available very faster, without making the process without making the process more tedious or easy. So these are the major takeaway from ICH, and by 2040, ICH goal is that it will be expanding the membership and will be creating a more inclusive stakeholder community and provide cooperation and collaboration, and harmonization initiatives will be there for addressing various international health products, including not only pharmaceuticals but biotechnological products also. And not only in developed countries, but in developing countries also, implementation of this ICH guidelines need to be explained, trained, and implemented. So these are the references. You can find all this content in uh, this ICH website, that is www.ich.org. And I hope, I think we have taken optimum time, right, Kathy? Thank you all.
Yes,
Yes, so
ma'am.
I
Yes,
think I
ma'am.
was able to reach to the students. I hope the
Yes,
students were
ma'am.
able
Thank
to
you so
understand.
much. Yes, ma'am. Thank you so much for such an informative session. And our students have some questions, ma'am. May I?
Yes, 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 surely.
Ma'am, first question is, can a person other than ICH members suggest a topic for a new ICH guideline?
Yes, yes, yes. Can
Okay.
suggest. So pharmaceutical industry, all these topics, they are identified in various discussions where the regulatory agency and the pharmaceutical industry, they look upon, or in the discussion they identify that yes, what hurdle pharmaceutical industries are having, what issues they are facing, and depending upon the issues they are facing, and how it can be made like more feasible to the patient, more available to the patient in terms of the healthcare, right? In that terms, the patients, not only patients as a important stakeholder, but the public also can suggest.
Okay, ma'am. Okay. Next
Okay.
question is, what is the meaning of bracketing and matrixing designs in QW guidelines?
Yes, so bracketing and matrixing, actually the time was running, so I
Yes.
hurried through those slides.
Yes.
So bracketing design is nothing but it is particularly for testing or stability testing program where it is reduced design,
Okay.
right? So formally, actually, real-time testing stability program studies which we carry out, that is called as full design. Full design means
Okay.
the testing it is carried out at all time points. But bracketing and matrixing, they are called as reduced design. And reduced design meaning testing it is not carried out at all time points. We can skip different time points, right? So that is the meaning of
Yes.
bracketing and matrixing design, which is reduced design. It is not a full study design.
Yes. Okay,
I hope
ma'am.
it is clear.
Uh, yes. Yes, ma'am.
Yes,
Ma'am,
yes.
next question is, what is the difference between biotechnological and biological products?
Yes, so biotechnological and biological products. So as we see the guidelines and all, I think the students must have referred. So biotechnological derived products are these DNA based products and which are derived from the like animal or human origin using like this different vectors and all, where viral safety evaluation is very, very important, right? So if you compare this biotechnological product with normal pharmaceutical product, then like manufacturing will be totally different from the pharmaceutical product, quality evaluation will be totally different, and the stability program will be also totally different from the pharmaceutical product. And biological products are nothing but the like I think students are aware about the blood products like
Yes, ma'am.
plasma. Yes, yes. So these are all the examples of the like biological products which are there, and specific biological products which also includes this antibodies and all.
Yes, ma'am.
Okay,
Ma'am, next
yes.
question is, what is the difference between generic and standard drugs?
Okay, so generic and standard drugs, I think the students are aware. Generic word, it is used for the formulations where it will be different from the company who has invented. So when the formulation is available first time in the market, it is called as the inventor formulation, right? So the formula, like certain excipients can be changed in the formulation, but the use remains the same, right? The formulation can be also the same. Like say, for example, in the market, I can give you an example. Like say, tablet formulation is available, which is launched by that particular inventor, and after this patent like expiry or like that, right? A generic can be filed, right? And generic can be filed when the patent which has been done by the innovator company is going to expire, right? And generic can be filed where they should not infringe the formula of innovator or the inventor. And innovator formulation and generic formulation, how it differs? They use the clinical indication, the formulation type, that is, say, there's a tablet formulation that remains the same. But certain excipients and all those things will be changed. And for generic, you have to only comply, or you have to only assure for bioequivalence study. That is a major important thing, that when you want to launch a generic formulation in the market, you need to perform bioequivalence study. So there is no need to perform those like detailed non-clinical study and clinical study, which are listed in M4 and M5 module. So along with M3 quality information for generic, you need to perform bioequivalence. So bioequivalence study for generic has to be compared with innovator formulation, and if it is found to be bioequivalent, right, with respect to results, then they can launch that particular or put that particular generic formulation in the market. So that is how generic formulation differs in terms of formulation and availability in the market from innovator formulation.
Okay, ma'am.
I hope this is
Ma'am,
clear.
next question is stability
Yes.
testing and stress testing are similar or not?
See, stability testing, when it is carried out at normal temperature, right, that is long term stability data, right, stability program. So, which is carried out in long term duration. So, that is called as real time stability studies. Depends upon the
Okay.
zone, right? It depends upon the
Yes.
different zone. That different temperature and humidity has to be selected with respect to the zone, and then stability studies has to be performed, right? But accelerated studies means you are elevating or you are increasing the temperature, you are increasing the humidity, and then you are checking whether that APR or formulation is stable or not. That is the
Yes,
difference between accelerated. So, why we perform accelerated stability testing? So, as to predict that, say, up to shelf life, when you perform this real time stability studies, that is long term stability studies up to 1 year, right? Then, say there is no change in the any parameter, that is physical or chemical parameter of the formulation, right? There is no variability or no changes has been observed. But same stability testing which we perform in accelerated condition where temperature has been increased, so as to check that over the period of time, like after 1 year or after 2 years, right? Under normal environmental conditions, like if you see the normal environmental condition, there will be fluctuation in temperature, fluctuation in the moisture content, oxygen level, right? So how it is going to impact, so that is being extrapolated by performing accelerated. An accelerated stability study, it is always performed for shorter duration. Why? Because when you perform for a short duration at higher temperature, there is likelihood that API will undergo what? Degradation, which is observed at long term after say 1 or 2 years. So accelerated stability testing are nothing but forced degradation conditions which are used, and that
Okay.
are
And ma'am,
uh,
last question is, ma'am, terminologies like drug products and drug formulation are same or not?
so in ICH, I think students must have referred the guidelines. So drug substance and drug product. So drug substance, it is specifically used for API, active
Yes.
pharmaceutical ingredient, that is drug substance. And they write it as DS, capital DS. And
Okay.
drug product, it means it is a formulation.
Yeah. Okay.
Drug product, that means it is formulation. So
Okay.
formulation can
Okay.
be anything, that is tablet or all those things, right? Capsule, injections,
Yeah.
all those things.
Okay, okay. Thank you so much, ma'am. It's a great pleasure having a speech from you, ma'am. Dr. Subhas University is very thankful to you, ma'am. Thank you so much.
Thank you so much again, Khyati, and thanks to the like School of Pharmacy and Subhash University also for giving me this platform. And I think the topic was interesting enough for the students,
Yeah,
isn't
sure,
it?
sure.
Actually, ICH,
Yes.
they find it as a boring topic because they find
Yeah.
it that it is full of guidelines.
But
But just
you make it
explain them
interesting, ma'am.
that they need not remember all the guidelines, because
Yes.
guidelines are put up on the website. They can refer it. Just they need to understand the scope of guideline, that why this guideline has been framed and what is the importance of the guideline. Just remember that much only.
Yeah.
Right.
Thank you so
So
much, ma'am.
yes, yes, yes. Okay. Thank you, Kathy. Okay.
Yeah. Okay. Okay.
Bye. Now we can leave,
Bye.
right?
Yeah.
Okay. Okay. Thank you.